Okay, welcome back. And I again need to reconfirm from you people that whether you are able to listen me and see me. So if that part is okay, so we can go ahead. Welcome back. So kindly, kindly just confirm me that you are able to listen me. Okay, yes. So Yash and Ankit has confirmed that they are able to see and listen me. So now we have completed our exercise for a this two alleles and three alleles. Now we will try to extend that knowledge for if say for example we are having k alleles, then what we will do. So, from the above example, you might have seen that the number of alleles are taken here for making these grouping variables, right? And these dummy variables for different alleles, okay? And genotypes are written here, and these are the number of genotypes for a particular genotype, number of individuals with a particular genotype. And this is how we multiply each number of genotype with the dummy variable, isn't it? Now say for example there are k alleles. So one generalized formula you must understand here is that if there are k alleles, then the number of possible of genotypes in a population will be number of genotypes are equal to k into k plus 1 divided by 2. So say for example there are two alleles. So number of genotypes will be 2 into 3 divided by 2. So it is equal to 3. If there are 3 alleles, then it will be 3 into 4 divided by 2. It will be 6. If there are 4 alleles, then it will be 4 into 5 divided by 2. So it will be 10 genotypes. So basically this is how by using this formula we can calculate that what will be the total number of genotypes for k alleles. So the last number will be k into k plus 1 divided by 2. Okay. And in a line we have introduced already this thing that the sum of all the alleles, the dummy variables, this sum should be equal to 2. Okay. So if 2 has been taken by A1, then all other alleles will be 0. If say for example this A1 and A2 has taken 1 value, then all other alleles will be 0. If all other alleles are 0, then the last allele has to take the value 2. So this is how our alleles are being distributed in the population. So this will make your A1 genotype, this will make your A1 A2 genotype, this will make your AK genotype. And these will be number of individuals with A1 genotype, these will be the number of individuals with A1 A2 genotypes, and likewise these will be the number of individuals with AK genotype. Now, their sum will constitute your genotype pool, or the total number of individuals will be the total number of genotypes. If you add these all, it will be the total number of genotypes in the population equal to the genotype pool. And for each allele, if you sum all these, so these will be the number of A1 allele in the population. These are the number of A2 allele in the population. This is the N dot, and this is the number of AK allele. That is because there are K alleles in the population. So if you add these all, so it will be equal to twice of this N dot dot. Okay. So if we have the same thing I have shown here, that if we have k is equal to 2, the number of genotypes will be like this. If we have k is equal to 3, then the number of genotypes will be like this. Okay. So here these k alleles A1, A2, AK can be taken as k dummy variables that can take any dummy value 0, 1 or 2, such that in each row A1 plus A2 plus AK will always be equal to 2. Since a diploid individual at a time can only have two alleles out of all possible alleles, right? Now, how we will calculate these genotypic frequency? So this n dot dot will be equal to the sum of all the genotypes present in the population, and we have used this generalized formula for individual. Okay. Similarly, for say for example, this is we want to find the probability of A1, then the number of individual with A1 genotype divided by total number of genotypes in the population will give you A1. So similarly, this AK will be an AK, and sum of all these probabilities will be equal to 1. Okay. Now for allelic frequencies, you have to add all the alleles individually in the population will be equal to n dot. Okay. And this will be the probability of occurrence of a particular this allele. Okay. So say for example, PA1, it will be NA1 divided by n dot, this NA2 divided by n dot, and this NAK divided by n dot. And if you add all these up, okay, so this will constitute your uh, allelic frequencies for the K alleles. So because now you have the background that how we have calculated for the 2 alleles, how we have calculated for the 3 alleles, you can very well imagine in your brain that how we can calculate the allelic as well as genotypic frequencies for a locus having k alleles. Okay, that is any number it can take. It can take 5, 6, 7, 8, 9, 10, or 100 or 1000 allele. Okay, so as the number of alleles will increase, of course, we will be requiring software and computers to solve these equations. Okay, now we have completed with the autosomal locus, isn't it? And when we have started this topic, I have told you that there are allosomal locus also, that is the sex chromosomes, right? So now we will try to understand that if there is a sex chromosome and there are one allele, two allele, or three allele, or k alleles are there, so how we can go ahead, right? So an allosome is a sex chromosome that differs from the ordinary autosome in form of size and behavior. Okay. So I will try to give you a basic about these allosomes, these sex chromosomes, okay? And then we will try to understand the rest of the things, right? So all diploid organisms with allosome determined sex get half of their allosome from each of their parents, isn't it? Now in mammals, females are XX and they can pass along the X chromosome, and since males are XY, they can pass along either X or Y. For a mammal to be female, the individual must receive an X chromosome from both parents, whereas for male, the individual must receive an X chromosome from their mother and Y chromosome from their father. Okay. Now, in humans, if you see, X chromosome carries around 1500 genes. Okay. X chromosome carries 1500 genes, and more than they any other chromosome in the human body. Uh, Understand this thing, that X chromosome is having maximum number of genes in the as compared to other chromosomes in human body, and most of them code for something other than female anatomical traits. It is not the female anatomical traits. This is a misnomer. Okay. As such, if you think, you might think because it is a sex chromosome, it might be producing something that is related to female, but it is not so. They code for something other than female anatomical traits, whereas the Y chromosome carries about 78 genes. Okay, only 78. And most of the Y chromosome genes are involved with essential cell housekeeping activities and sperm production. And only one of the Y chromosome gene, that is SRY gene, okay, is responsible for male anatomical traits. So this is the gene which define your male anatomical traits. I'm just giving you this background for understanding the how this structure of these genes are being made. Okay, and I am relating it with the humans, so it will be more pertinent, and you will be able to understand about the allosomes in particular. So recombination between X and Y chromosome proved harmful. So if there is a meiosis, there are no recombination between an X and Y. Okay. and it resulted in males without necessary genes formerly found on the Y chromosome and females with unnecessary or even harmful genes previously found only on the Y chromosome. It should be X. Okay. As a result, gene beneficial to male accumulated near the sex determining gene. Okay. So, and recombination in this region is suppressed in order to preserve the male specific region. Okay. So, recombination in the male specific region is not there and it is suppressed. Over the time, Y chromosome changed in such a way as to inhibit the areas around the sex determining genes from recombining at all with the X chromosome. As a result of this process, 95% of human Y chromosome is unable to recombine and is called as non-recombining region of Y chromosome or male specific region. Okay, so either it is NRY or MSY, and it is the 95% of the region in the Y chromosome that do not recombine with the chromosome. Okay, only the tip of the Y and X chromosomes recombine. Okay, so this is important for you people to understand that there are only few genes which are able to recombine in case of Y and X chromosome. Now, the tips of the Y chromosome that could recombine with the X chromosome are referred to as pseudo-autosomal region. And why I am telling you this thing? Because this is generally being asked in competitive examinations. Okay. So if somebody say, is there recombination between Y and X chromosome? Of course it is there, but it is only in the 5% region, and that too in the pseudo-autosomal region. Okay, that is known as PAR. So this is the basic structure of a Y chromosome, and you can see that at tip there are PAR 1 and PAR 2 region, and rest of the region are MSY. Okay, and these regions are not taking part in the process of recombination. Okay, so now we will try to understand that how these uh, allelic and genotypic frequencies in case of your sex-linked genes or allosomal locus can be determined. Okay. So first is two alleles. Say for example, it is a allosomal locus and there are two alleles, right? So the genetic structure of population for a trait governed by sex-linked gene having two alleles have been given below with the help of illustrations and examples. So I am taking one of the example of X-linked hypohidrotic ectodermal dysplasia in dogs. Okay. So X-linked hypohidrotic ectodermal dysplasia, which is known as XLHD, is a genetic disorder characterized by abnormalities in ectodermal derivatives such as sweat glands, hair, and teeth. In animals, the highest number of cases has been reported in dogs, which show characteristic congenital alopecia. So what will be there? There will be congenital alopecia and develop abnormalities in the shape and number of teeth. Okay, so its inheritance is X-linked recessive, right? So say for example, how it is being governed is that I will just reduce this thing 150. So this is the probability of being born with XLHD from the mating between a female carrier and a normal male. Say for example, it is the female carrier. So this particular allele is responsible for XLHD. It mates with a normal male. So these are the two alleles formed by the female. These will be the alleles formed by the male. So ultimately 25% normal female, 25% normal male, 25% female carriers and 25% affected male will be produced from this mating. Okay, so this is how this is being transmitted in the progeny, right? Now, what are the variants? So, male only have one X chromosome, whereas female have two. Therefore, the possible genotypes differ by sex, right? So, female dogs having AA genotype and male with A genotype will not have excess LED, whereas female dogs with A/A genotype will not have this disease but are carriers. If a carrier female is bred to a normal male, all female puppies will be normal, but 50% of them will be carrier. Among male puppies from this cross, 50% will be normal and 50% will be affected. Female dogs with small a genotype and male dogs with A genotype will have this particular disease, hypohidrotic dermatitis. So these are some of the figures of this hypohidrotic dermatitis, and when I was reading this particular paper, so it has been shown that not many doctors know about this thing. When a dog comes to them, they used to treat it, okay, using the medicines, but it is a sex-linked disease which occurs congenitally and it is genetic in origin, and the doctors and the patients must be informed about this thing. Okay, so basically, if you see this AB and CD, so AB is a Pekingese crossbred at 9 years of age affected by X LHD, showing the characteristics of alopecia region. You can see the alopecia regions. Okay. C is the oligodontia and conoid teeth. Oligodontia and conoid teeth you can see. And note that there is a mesio version of three of the upper incisors. Okay. There is a mesio version and labio version, okay, labio version of the lower incisor. So, and D is the histological section of area of alopecia showing hyperkeratosis, normal collagenous fibers, and absence of piloglandular units. So, this is being taken from one of the paper of this 43 is Maura et al. 2019. Okay, where they have reported this particular disease. So now we will try to understand in a population that what would be the this allelic and the genotypic frequencies for this particular type of disease. So, example, a sample of Pekingese crossbred dog population of 1000 dog was screened genetically for the presence of X-linked hypohidrotic ectodermal dysplasia alleles. Okay. Now, following genotypes were mapped to different dogs in the sample. So there were 200 females with this AA genotype, 215 with the this A and small a, 50 with the AA, that is having this disease. Whereas in males, 400 were this type, so normal, and 100 were having this disease. Okay. So calculate the gene and genotypic frequency of this sample from the population. Now we will try to understand that how we will be calculating the gene and genotypic frequency in this population. So the gene and genotypic frequencies in case of sex-linked traits are calculated separately for male and female dogs. So here the main point is that the female act as a diploid organism. So the main funda is what I found to make it easier is that simply you consider female as a diploid organism, whereas the male act as a haploid organism. Okay, because not all the genes of those are present on the X are present on the Y. So because it is not present on the Y, the male organism for this particular gene act as a haploid organism. Okay, so there you now recall that when I was using a dummy variable, the sum of all the alleles must equal to 2. But in case of haploid organism, it will become 1 instead of 2. Okay, so we will be having the female tabulation as it is, as you do for the autosomal allele. Okay, same thing 201102, these are the genotype, these are the numbers, you calculate them total allelic frequencies and the genotypic frequencies will remain same as that of this. Okay, but the difference will be basically in the male population, whereby now you see that A and B, the sum of the two will be equal to 1 instead of 2. Okay, because the male population will be acting as a haploid organism, and the genotype will be either A dash or the B dash, isn't it? Then the number of these are the numbers, so it will be the total. And one more basic thing here is, then when we were doing earlier for the diploid organism, because it was diploid, the sum total of all the alleles was twice the total number of genotypes. But in this case, it will be equal. That is, if you add this with this, so it will be equal to 500. Okay, it will not be twice. So what we are doing here is, here two alleles A and B are two dummy variables that can take any dummy value among 0 and 1, such that A plus B will always be equal to 1. Since male individuals will act as a haploid individual for X-linked traits, therefore they can only have one allele at a time. So these are some of the generalizations, because other part of the genotype is not having anything, so it will be dot and dash. So it will be equal to n dot dash divided by n dot dash. So you can have these values. So similarly, for allelic frequencies and genotypic frequencies, in case of these your males will be same. Okay, that is the genotypic frequency and allelic frequency in case of males will be same. Why? Because you see here that it will remain same. This is 400, this is 400. Okay, and this will come out to be, for the A allele it will come out to be 400 only, and this will be 100 only, which is the total, which is equal to the B. So the genotypic frequencies and the allelic frequencies in case of male will remain same, and which will be equal to 400 divided by 500, 0.8, and 100 divided by 500 is equal to 0.2. So basically, in case of these allosomes, the males you have to calculate it separately. Females will go as it is, those of the as we have done for the autosomes. Okay, but in case of male, the gene and the genotypic frequencies will be the same, and in case of dummy variables, it will, their sum should be equal to 1. Okay, now if I take k alleles, okay, so I have made the generalization for the males only, because female will be the same generalization we have used earlier. Okay, and here the allelic and the genotypic frequencies will remain the same, and it will be like this. Okay, now I will just try to uh, finish this chapter by telling you the last part, the basic generalization for multiple autosomal loci with multiple alleles. That is, till now we were talking about one loci, either two alleles or many alleles, allosome, autosome. Now I will be talking about multiple autosomal loci with multiple alleles. Now we will try to deduce the genetic structure of population for a trait affected by different genes at multiple autosomal loci and bears multiple alleles. So for that I will be taking one of the illustration, and that illustration is allelic dominance and hierarchy of individual loci for coat color in dogs and cats. Okay, it is being given by Korek et al. Okay, in 2019. From that paper I have taken it, this example. So if you see these locus, so I will reduce this thing. Yes. So inheritance of basic coat color loci in dogs and cat are like this. There are actually three locus are there. One is the E locus, other is K locus, other is A locus. So there are basically three genes. If you see the E locus, you will see that there are different alleles are there. And what are those? Capital E, small e, E, M. Okay, so these are the three alleles, different alleles. Then for K locus, there are different alleles. And for this A locus, there are different alleles. If you see these, so for E, there are three alleles are there, and this is the order of dominance. That is, EM is dominant to E and E is dominant to small e. Similarly, for this K is dominant to this KBR, and this is dominant to small k. And this for A locus, there are four alleles are there. So these are AY is dominant to AW is dominant to AT, and it is dominant to A. Okay. So basically all these interact with each other also. So if this is the genotype. So these will be the outcomes. But if this is the genotype, then it will be decided by the K locus. Okay. So if this genotype plus these K locuses are there, so these will be the two phenotypes you will be getting. Okay. And if this K locus and this is this, then the picture of the A locus will come into play, and these will be the different colors, those you will be observing. So in total 1, 2, 3, 4, 5, 6, 7, 8 types of phenotypes you will be getting from different genotypes, right? So basically now we will try to understand that how we can deduce a relationship for this particular many loci and this multiple alleles. So now if we know that K alleles, so there will be the number of genotypes will be like this. So for E and K we have 3. So total number of genotypes will be 6, whereas for this another locus A, the number of alleles are 4, that is K3. So that will be equal to the number of genotypes will be equal to 10. So the total number of genotypes considering 3 loci at a time will be 360. If I consider all these 3 at a time, so there will be different number of genotypes will be 360 type of genotypes. Okay. So basically a generalized formula for calculating the total number of genotypes for 5 loci. Okay, each having K1, K2, K alleles. That is, number of genes here will be 5. Oh, xylem, sorry, xylem. Different notations I am using so that these can be differentiated from one another. And there will be K1, K2 and K omega alleles. Okay, and consider that they assort independently. So the number of genotypes will be equal to K1 into K2 into K3 into K omega, then into K1 plus K1, K2, K3 plus 1 into this, and up to K omega into 1, divided by 2 power number of genes you are considering. So if in above illustration I take, so how many K1 are there? 3. K2 are 3. Then for third A locus there are 4. Then you have to multiply with the plus 1. Then everything you add 1, that is 3 plus 1, 3 plus 1 and 4 plus 1. Multiply them. And 2 power number of genes, that is the number of genes are 3. So you put into this formula, so you will get 360. Okay. So the total number of genotypes in this case using this formula will be equal to 360. Okay. So now solving a hypothetical example based on the above illustration with 360 genotypes is beyond the scope of this lecture. However, one can very well accomplish this using computer softwares like Excel. However, we can deduce a generalized methodology to understand how gene and genotype frequency can be calculated for multiple loci with multiple. So I have used this table. It is not meant for you. Okay, that is for undergraduate level. It is more than enough what we have used earlier. But it is just for the curiosity of the students. I have just added this thing to your these lecture notes. That is using these. There is nothing but what I want to tell you here is that these are the different alleles for the different genotypes. Okay, this will be the 5 will be the last genotype. That is, this is the first genotype, first allele, first genotype, dot allele, first genotype, K1 allele. Then this will be the some in between genotype, this allele. Then 5, this xylem allele, xylem gene, A1 allele, xylem gene, A dot allele, then xylem gene, AK omega allele. Okay. Similarly, this is how these are the different number of individuals for different genotypes will be there. Okay, that depends on how many genes and how many alleles you are in your system are there, and these will be the different number of alleles you will be having. Okay, that again depends upon how many individuals you have, and again you have to tabulate them, and accordingly by using these formulas you can calculate the allelic and genotypic frequencies for any number of genes with any number of alleles in a population. Okay, so in summary, in this lecture note, we understood the basic concept like biological organization, population and its types, Mendelian population, Methodologies to calculate the gene and genotypic frequencies in a population have been explained for autosomal and allosomal locus. Tabulation and methodology using dummy variables to calculate gene and genotypic frequency for single locus, two allele system, okay, and to that of multiple locus and multiple alleles have been undertaken. So this last part I recommend it for those who do PG or PhD work. Okay, they can take up. Otherwise, just for understanding, I have put this that it is not that we can do for only for one or two alleles or only for one or two genes. We can take any number of genes with any number of alleles to deduce the genotypic and the gene frequency in a population. Okay, so that's all from my side, and I think because there are no questions, so this ends our this lecture, and soon I will be publishing these lectures, and after publication I will be sharing these on the websites, so they will be readily available to all of you, and you can go through them whenever you have the time. So this is all from me today, and those who are watching, thanks to them for being there to listen me. Thank you very much.